But yeah, so we're gonna talk about both SL and HL, but I will indicate when we start talking about HL stuff, and then we'll take a 5-minute break in the middle after I finish D1.1, and then I'll send out another ping saying that I'm starting with protein synthesis for SL, and then I'll indicate when I start talking about protein synthesis for HL as well. Okay, so this is, I think, trickier for HL, but it's fairly difficult for SL also, I think. I mean, generally speaking, for most people, I think it's okay. But yeah, so we'll just talk about, I'll just start. I don't know what I was saying. Anyway, DNA replication. So, DNA replication is the synthesis of new strands of DNA with precisely the same base sequence. Now, this is important. Precisely the same base sequence as the original strands. The HL will... So, I'm going to do SL, HL, SL, HL, if that makes sense. Because I'm going to talk about DNA replication and I'm going to talk about protein synthesis, but then SL, HL, SL, HL. I'll indicate when I transition to HL, though. Yeah, so that's I think that's the definition in the Oxford study guide or the Oxford textbook. It's the same. But I think it's a fair enough definition of it. It's the synthesis of new strands of DNA, so you're making new DNA, with precisely the same base sequence as the original strands. That's the important part because you're making, essentially you're producing two DNA molecules from one DNA molecule. So it's kind of like cell division. But yeah, so DNA is replicated before cell division because DNA is in the nucleus of cells, obviously. But it's replicated before cell division so that both daughter cells have the entire genome because you don't want to lose that. That's where DNA conservation comes in. We keep, in a body, you want to keep the same DNA sequence. In multicellular organisms, cell division is part of 3 different processes, and this is specific to multicellular organisms. For unicellular organisms, it's only part of the reproduction slash replication part, not really the growth or replacement of damaged tissues. Now, those two are where cell division comes into part, but reproduction is also one of them, where cell division essentially helps with the creation of, basically to provide cells that will develop into gametes. So the reason this is important is because we're trying to say that DNA replication is an example of continuity because you want to pass on those base sequences unchanged from cell to cell, and from generation into generation, which is why DNA replication essentially just is the way our body remains how it is throughout generations, very generally, in the most general of terms. Okay, so at the bottom here is just an example. That's a DNA molecule. After replication, we'll have 2 DNA molecules that are the exact same product. Okay, next. DNA replication is semi-conservative. Now, what that means is replication starts with the separation of a parent DNA molecule. So that parent DNA molecule, everyone should know that DNA is a double helix. That DNA molecule has 2 strands, which get separated at the beginning of replication by breaking the hydrogen bonds between the bases of a DNA strand. And then each single strand is used as a template for the assembly of a new polymer of nucleotides, which then creates 2 different DNA molecules. The reason it's called semi-conservative replication is because each of the DNA molecules produced has one new strand and one strand conserved from the parent molecule. Hence the semi-conservative. It's saving half of it. So you're saving half of the parent molecule and then you're adding a new strand to each DNA molecule. Okay. Just to make this a little bit less just me speaking all the time. What are the 4 bases in a DNA molecule? Or in a nucleotide. What are the 4 bases? Well, yes, ATGC is the simple answer. I want the full names, and please spoiler your answers. In order to spoiler them, do this. Hold on. That. In order to spoiler them, just add 2 bars on either side. I mean, it's in the textbook, so I would assume you need to know it. But yeah, so that is right. I'm just gonna, yeah, that is right. It's adenine, thymine, cytosine, and guanine, or guanine, I don't know. That, guanine. Guanosine, I'm not even sure what it is. I don't think we need to know it as part of IB. And Lake, the thing is, I asked what are the 4 bases for a DNA nucleotide, so you don't want to add uracil there because they will mark you down for that. Because this question was specific for DNA nucleotides, not RNA. Okay. Yeah, so that's semi-conservative replication. Does everyone understand semi-conservative replication? Or does anyone need a better explanation of what it means? Ignore my Coke can. Someone's still typing, so I'll wait a second. Oh, there. It says students should know the name of the nitrogenous bases. I mean, I don't think it's too hard to know, to be fair. It's just adenine, guanine, cytosine, and thymine. I said that already. Adenine, thymine, cytosine, guanine. Basically. But yeah. Okay. Does everyone understand semi-conservative replication? Yes. Perfect. Okay. Now we're going to talk about some of the more tricky things. Helicase or helicase, or however the hell you want to say that name, and DNA polymerase. So, a few things you need to know before we start talking about this. What's a replication fork? So a replication fork is the site where a parent DNA molecule is separated into 2 single strands. So in this case, that would be the fork. That's where it's being separated at the moment. Each of which is a template strand. So when you're talking about DNA replication, yeah, this is just SL stuff. When I talk about HL stuff, I'll say that I'm talking about HL stuff. But for SL, all you need to know is that it's just DNA polymerase. So when you're talking about DNA replication, both of them are template strands, not one of them. Both of them are template strands. The difference, we say, is leading and lagging strands, if you want to know that. Hold on, I have a knock on my door. Let me just check. Anyway, okay. Yeah, you just need to know helicase and DNA polymerase for SL DNA replication. Right, guanosine triphosphate. That's right. That's right. But yeah, you don't need to really know that. You just know it as G3 phosphate, I think. That was last week. I'm not going to talk about cell respiration, Calvin cycle. What was I saying? Right, so both of them are template strands. We talk about them in the sense that one's a leading strand and one's a lagging strand. But yeah. So the replication fork gradually moves along the parent molecule. Obviously, if this is the fork, as more nucleotides get added, it just moves across further. And essentially, the entire process occurs due to enzymes. And for SL, you need to know two enzymes, helicase and DNA polymerase. So helicase is essentially what unwinds the double helix and then separates the two strands by breaking the hydrogen bonds between the nitrogenous bases. So between C and G and T and A, or adenine and thymine and cytosine and guanine, there are hydrogen bonds which are broken by the helicase in order for them to be separated into individual strands and then create new DNA strands. That's the job of a helicase. DNA polymerase is what links the nucleotides together. So does everyone know what a nucleotide is? You should know this from nucleic acids, from I think. I don't even know how they divided it this time. But does everyone know what a nucleotide is? Okay, good. Does anyone, actually, I should ask, does anyone not know what a nucleotide is? Okay, I'm gonna assume no answer means everyone's good. Yes, so it's, yeah, probably easier is if I just explain it. A nucleotide is a phosphate bound to a pentose sugar, which means 5 carbon sugar, with a nitrogen base. Those nitrogen bases are ATGC. So each nucleotide is attached to the template strand by hydrogen bonding, by complementary base pairing, through using DNA polymerase. That's what links the nucleotides together when they're being attached to the template strand. Everyone should know complementary base pairing. If you don't, we talked about ATGC. So adenine and thymine are one complement, and guanine and cytosine, or guanine and cytosine, however you pronounce it, is one pair. So at any point in time in a DNA molecule, these are the only pairs that will form. You will not have a cytosine binding to a adenine or a guanine binding to a thymine or anything like that. It's always AT and GC. So complementary base pairing ensures that the new strands are assembled identically. So then obviously it's going to be the same. So as you can see here, ATGTC is the original 5, like the beginning 5 nucleotides or base sequences. And then the ones that get added are the complement or what would have been on the other strand. And it's just replicated. AT has 2 hydrogen bonds and C and G has 3 hydrogen bonds, yes. Okay. Do we understand helicase and DNA polymerase for SL? Okay. The only thing I'm not discussing today is PCR, because I want to do, I've said this before, but I want to do an experiment session where I talk about every experiment that you need to know. So I want to talk about PCR and what else? What did I say last week? Chromatography and gel electrophoresis and other such experiments all together in one, if that makes sense. Because I think that'll be easier than me sitting here and explaining PCR for like half an hour. If that's okay. But if you guys really, really want to know right now, I can explain it off the top of my head. But yeah, you can let me know. But until then, this is insane. This is insane. I was talking about PCR is in SL also, so it's both. PCR is a general topic that you need to know, which is why I want to do experiments all together, because all the experiments are both SL and HL, so it helps if I do it together. Is that good? Okay, I'm going to talk about direction of replication. So, all the nucleotides in a single strand of DNA are oriented in the same way, and this is because the strands are assembled by DNA polymerase, which always adds the phosphate of a free nucleotide to the deoxyribose of the nucleotide at the growing end of the chain. So essentially, it won't add it here. Any nucleotide that's added will be added here, where that phosphate group will be attached to the deoxyribose of this nucleotide. So that and that get joined. So whenever replication is happening, it will always happen in this direction, never the opposite, because the free nucleotides are what get attached to the deoxyribose of the groups, but not the other way around, if that makes sense. So it's always top to bottom. Yes, this session is being recorded. What else was I going to say? Yeah, so the direction of replication is from 5' to 3'. Now, everyone should know what that means from nucleic acids. I don't want to re-explain that, but everyone should know what that means. 5' to 3' is the direction of replication, because the phosphate group is, well, I am explaining it. I don't know why I said I don't want to explain it. I literally put it on my slide. 5' is the side of a nucleotide, and the phosphate group is the 5' and the deoxyribose is the 3'. Therefore, the direction of replication is from 5' to 3' and not from 3' to 5'. Does that make sense? I literally did not want to explain it, but now it's on my slide, so I will explain it, I guess. I mean, this directionality was in the study guide, and it says it's part of SL. If you don't need to know it, okay. But you should know replication happens from 5' to 3', essentially. Okay? Is this the last SL thing? This is the last SL thing for replication. Other than, it is a +1 in the paper 2 section B. Trust. Agree. Okay, this is the last SL thing for replication. And then we're going to talk about HL replication. So, there's the leading strand and the lagging strand. Now, I don't know if I did not include a picture of it, but let me put a picture in the... Never mind, I'll just go back to this picture. So, there's the leading strand and the lagging strand, right? But the difference is, in the leading strand, which is 5' to 3' oriented and not 3' to 5' oriented, RNA primer is assembled at the start, and therefore the DNA polymerase can move in the same direction as the replication fork. I know it says polymerase 3, but that was just because it said that in the textbook. But you, as SL, you don't need to know that it's DNA polymerase 3. If you just say DNA polymerase, it works as well. But because this is an SL HL thing, HLs need to know this. So the RNA primer is assembled at the start of the leading strand, and DNA polymerase 3 moves in the same direction as the replication fork. So if I go back to this picture, on the leading strand, it will be moving on the same direction. So it will be moving this direction. And with the replication fork, that means that it's going to stay continuous. It's going to be continuous replication rather than discontinuous replication, which means that it forms a single new strand of DNA rather than segments, which is what happens on the lagging strand. So in the lagging strand, DNA polymerase 3 is working away from the replication fork. Because obviously it has to move in the 5' to 3' direction, which is the opposite for the lagging strand. And then, so essentially for that, replication happens in segments where as the replication fork is moving along, primer will get added to certain segments, and DNA polymerase will start making strands, and then it will happen again a little bit further down the lagging strand. But then what happens is, as DNA polymerase does its work, it'll meet the previous already made strand. So essentially, let me go on this. For example, although this is not really good, this picture is not the best replication of this, but if your primer is here, for example, because the fork is going this way, right? So if in the beginning the primer attached itself here and formed a DNA polymerase, the DNA polymerase formed a strand, and then as the fork moves along, the primer then attaches itself here and then goes along and builds one, but then it will meet the previous primer, and then there will be a disconnect between this strand and that strand, which that disconnect leads to it being in fragments, and those fragments are called Okazaki fragments. And replication has to be reinitiated after they meet the previous RNA primer. And therefore, replication here is discontinuous. So essentially, all of that yap is just to say that on the leading strand, replication is continuous. On the lagging strand, replication is discontinuous. On the leading strand, replication happens in the same direction as the replication fork. And in the lagging strand, the replication happens in the opposite direction of the replication fork. That's essentially all you need to know. I did explain it because I think it can be a question in paper 2 section B where they say explain replication. So you want to explain this thing where, because it has to move in the 5' to 3' direction, it's the replication is continuous / discontinuous in the leading / lagging strand. Section B is not common for SL or HL, but it's because they'll give you, but there will be some SL questions in the HL paper as well. Okay, does everyone understand is it AHL in the guide? It said SL on my thing, so I don't know. I thought it said SL. I thought that was, okay. I might be wrong. This might be HL, guys, sorry. I was looking at the Oxford study guide, not at the actual, subject guide, which is why it might be wrong. Anyway, the SL kids, if you don't need to know this, don't know it. It's okay. I'm looking at the Oxford study guide. I thought it said SL, but I might just be blind. It's fine. If you don't need to know this as SL, just ignore everything I said. Follow what the IB study guide says. If the IB guide says that you don't need to know it, you don't need to know it. I've just explained it for the sake of explaining it. So this might be an HL thing as well. Okay, so SL technically ended here, is all I'm going to say. SL ended at helicase and DNA polymerase. Anything after that, I said, is all HL. Sorry. If you guys want to stick around, the HL part is interesting if you care to understand. If you don't, then it's okay. Anyway, so, okay. Just to reiterate, direction of replication and comparing replication are both HL. The fact that it's continuous and discontinuous and the Okazaki fragments and stuff, that's all HL. That actually makes sense now, because I was like, last year for SL, they did not need to know this either, so I don't know why you guys would have needed to know this year all of a sudden. So that makes sense. Okay, sorry to anyone who's SL and just heard me yap for the last 15 minutes for no reason. Okay, so now we're going to talk about, we have been talking about HL, but we're going to talk about more HL stuff. And then we'll take a 5-minute break, and then we'll talk about protein synthesis. So if SL people want to leave, you can. You don't have to be here. If you care to be here, feel free. Up to you. Okay, so first, we're going to talk about the enzymes, and then I'll talk about the whole process. These are all the enzymes you need to know as part of DNA replication. The reason you need to know these is because it can very easily be a 8-mark question or a 7-mark question in paper 2 section B, unless they've changed what paper 2 section B is. I'm so lost in the new syllabus, sorry. But I think it should be the same. It can very easily be a 7-8 mark question where they'll be like, what are all the enzymes used in DNA replication? Or they can just be like, explain DNA replication. In which case, this is still a good enough organization to go by. So you need to know the enzymes. Yeah, that's why you guys don't get those 7-8 mark questions, because you can't know enough to write those 7-8 marks worth of stuff. You usually get 4-3-4, I think, if I remember correctly. That's your division. So yeah. Okay, so enzymes. First is helicase. I've talked about this already. It unwinds the double helix. Next is DNA primase. So this is what adds the primers, right? So when I talked about RNA primer over here, that's why this is HL. That makes more sense now. DNA primase adds the primer to start replication. A primer is just a short strand of RNA, which is why it's called RNA primer usually. Like the primer is called RNA primer instead of DNA primer or just primer. And then there's 2 types of polymerase that we need to know. There's DNA polymerase 3 and there's DNA polymerase 1. So DNA polymerase 3 adds DNA nucleotides to the lagging strand to form the Okazaki fragments, as I already talked about. It also adds DNA nucleotides to the leading strand to form the new DNA strand. But I just said it because it's more important in the lagging strand. Like, one of these enzymes is more important in the lagging strand than it is in the leading strand. Is it RNA primase? I don't know, man. It said DNA primase on the... See, in the Oxford study guide, it said DNA primase. I thought it was RNA primase, but because it said so in the textbook, I just assumed they changed how they say it. But if you write RNA primase... No, I'm wrong. RNA primase is used for transcription, not for this. I'm This is why. So RNA primase is used in transcription and not in replication. Don't switch them up. I think that's why. Actually, I don't know, man. It says, okay, simple. It's DNA. It says DNA primase in the guide. Just write DNA primase. That's as simple as I will say it. Just, it says DNA primase on the guide, just no DNA primase. What was I talking about? I was talking about DNA polymerase 1. So DNA polymerase 1 is the other type of polymerase that you need to know. That type of polymerase is what removes the RNA primer and replaces it with DNA. But it leaves certain nicks in when it's replacing it, because it cannot make 3' to 5' bonds. It can only make 5' to 3' bonds. Those nicks are sealed by DNA ligase, which basically seals it over, gets rid of the nicks, makes it a continuous strand of DNA. Okay, more practically speaking, on this diagram, which I lifted from the Oxford Study Guide as well, because that usually has really nice diagrams about things like this. So this is the leading strand, since the replication happens 5' to 3' on the 3' to 5' strand. This people get like this trips people up sometimes, where people will be like, oh, but doesn't it happen on this because it goes 5' to 3'? No, because the template strand will be 3' to 5'. The bonds that are made newly are 5' to 3'. So the template strand for replication will always be the 3' to 5', like will always be in the direction of 3' to 5', and the replication will happen in the 5' to 3' direction. Okay, so first step, we're going to start here. Helicase, this little thing, is what unwinds the double helix and separates these two strands. Those are two of the strands of the DNA. And then DNA primase adds short lengths of short lengths of RNA attached by base pairing to the lagging strand. So here, that's the primer, for example. Now, each of these primers is where DNA polymerase can start. So DNA polymerase 3 can only know where to start when DNA primase sets down the primer. After that primer is set off, DNA polymerase 3 will bind at the 3' end of an RNA primer, so obviously on this side, and then adds a DNA nucleotide and moves along the lagging strand away from the replication fork. Again, this is for the lagging strand, away from the replication fork, assembling a strand of DNA nucleotides until it reaches another primer. So for example, if this is one of the primers, it will go until it reaches this primer. For this one, it will go until it reaches this primer. For this one, it goes until the next primer, which is what Okazaki fragments are, because they're separated by the primers. Those primers are then replaced with DNA by DNA polymerase 1, which will get rid of this bit of RNA and replace it with DNA. But it is unable to do it's unable to completely bind them, because it cannot make a 3' to 5' bond. So there's a little bit of a nick left. That nick is sealed up by DNA ligase, which then forms the sugar phosphate bond and completes the strand of DNA on the lagging strand. On the leading strand, DNA primase adds a primer to the 3' end of the leading strand, so over here, and then DNA polymerase will start DNA polymerase 3 will start to bind and assemble a strand of DNA nucleotides, and this is continuous replication because they'll keep going. There's nothing stopping it until it reaches the end of the leading strand, where obviously, since there's no more nucleotides on the template side, there will be no more nucleotides to be added, and it just disconnects it. That was a lot of yap. Does that make sense to people? Is there any questions about any part of that at all? Because that was a lot of just information. We explained the lagging strand. Yes. So basically what happens is, right, so if you look at the leading strand, the primaries can only be attached on the 3' side, right? On like in the direction of a 3' end, right? So then that 3' end is here for this template strand, and here for this template strand. So on the leading strands, it's continuous because it goes uninterrupted. When it's on the lagging strand, since helicase is still doing its thing and the fork is still moving this way, what happens essentially is it starts in segments. So it starts, for example, here, it starts there, it starts here where all the primers are. That's where DNA primase will add the primer. As this fork is moving, more segments will be created. Those segments are then filled with nucleotides by DNA polymerase 3, but it can only do that in the 3' to 5' template direction, which means it can only create bonds in the 5' to 3' direction, which means that it can only move this way. And because it moves this way, there's small segments already created. And then once the DNA polymerase 3 reaches from one primer to another primer, it stops there because there's nothing more. It can't build past that since there's already DNA created there. Once it reaches those that RNA primer, it just continues doing it for other segments. But since there's RNA primer in between, those are fragments. Those fragments are called Okazaki fragments. Then those fragments become one when DNA polymerase 1 replaces the RNA primer with DNA, and then the DNA ligase actually completes the full bond. Does that make sense? This is all HL. Does the DNA polymerase 3 on the lagging strand... Okay, so here's how I'll explain this. DNA polymerase 3 will always create nucleotide bonds in the 5' to 3' direction, but the indicator on the template strand is from 3' to 5', because as we know, 3' to 5' is 3' and 5' are like ends. So if a strand is starting from 3' at the top to 5' at the bottom, the other strand has to have 5' on the top and 3' at the bottom, right? That 5' to 3' is the direction in which DNA polymerase works, but the 3' to 5' is the template strand direction. So that's why that's a little bit tricky in terms of saying that the nucleotides are added from 3 to 5. That's not how that works. This is the lagging strand where 5' to 3', that's the template. The darker strand is the template strand, and then the 3' to, well, this, or it'll be 5' eventually, that's the new strand. Does that make sense? Are we good? Okay. Explain the role of DNA primase and the RNA primer. No, it's okay. So basically, the DNA primase is adds short strands of RNA, which is called RNA primer. What the RNA primer does is tell the DNA polymerase where to start replication. Without the primer, the DNA polymerase is lost because it doesn't know where to start. Otherwise, it could get attached anywhere, right? In order for there to be a proper attaching point, that's what the primer is used for. That's why on the leading strand, the primer is only in one place, because the replication can be the replication can be continuous, so the DNA polymerase 3 can move down just by attaching here. Here, it has to be in segments so that the DNA polymerase knows to go in which direction, essentially. It's the point of the primer is to indicate where to start and which direction to go in. Okay, it is DNA primase, glad. But yeah, does that make sense in terms of what a primer is or does? No, it's okay. This saying that I explained it a couple times is fine. That's literally what I'm here for. The more number of times that I can explain in a different way that makes sense to you is the purpose of me being here, honestly. So it's okay. Any other questions? Because I know this is the main part of this entire, this chapter, because DNA replication, this is the main stuff. No, there's a little bit more, but this is what DNA replication is, right? So this is the main question they're probably gonna ask you and things like that. So yeah. Also, is this only in eukaryotes? No. DNA replication happens in prokaryotes as well. No? But you don't need to know that anyway. This is okay. Let me just explain this, right? There are differences between prokaryotic and eukaryotic replication, but this is the most basic. These are the basic principles. This thing is this exact model is from a prokaryote, but the basic is the same. They're not gonna say what is DNA replication in a eukaryote versus what is DNA replication in a prokaryote. They don't care about you knowing that. This is really basic bio, bro. Okay, that came out wrong. I don't think it's basic bio, but it's not. DNA replication differences between prokaryotes and eukaryotes is outside of what IB Bio expects from you, is the way I'm going to put this. Well, in terms of what bio is, it is basic bio. In terms of biology as a subject and as the wide scope of science it is, this is basic. But in terms of IB Bio, it's very basic. Post-transcriptional and translational differences are part of protein synthesis. I'll talk about that later. Okay, are the Okazaki parts fragmented on the lagging strand just because the actions of the enzyme polymerase 1 and 3 and DNA polymerase are delayed complete? Yes and no. Yes, because, actually, yes, basically. The point is that the DNA polymerase 3 can only work in this direction, right? So because it can only work in this direction, it will start working as soon as it can, and that happens as the fork is being created. So imagine if the fork's here, right? If it's on this level, and if it's only just been open to this level, the primer will just attach and start doing its thing, and then the fork will keep growing, and then the primer will attach again, and the primer will attach again as the fork keeps going. On the leading strand, it doesn't have to do that because as the fork keeps going, the polymerase can just move. On this direction, it has to go in this on the lagging strand, it has to go in this direction, so it'll just keep doing that as the fork expands. Does that make sense, B? I don't know if that was a good enough explanation. But essentially, because, yeah, it's just the movement of the fork is not a hindrance to the leading strand, but it is a hindrance to the lagging strand. Okay, they don't jump. There's multiple. Okay, there's multiple multiple molecules of that enzyme, right? So there's one DNA primase and one DNA polymerase 3 will be working here, and then another will be working here, and another will be working here. So they don't really jump. But I get what you're saying, and yes, it is in that general logic. Okay, in terms of understanding it, yes, the enzymes essentially jump forwards each time on the lagging strand. But what happens is, if it starts here, it will finish this and then jump here and then finish this until the primer and then jump here and then finish this until the primer and then jump here and then finish until the primer. There's nowhere else for it to go because the primer is already done. Does that make sense? Well, let me okay, just so I make sure I'm not giving you the wrong information, let me Google this. Okay. Yes, multiple DNA polymerase can be active on a single lagging strand. Let me actually double check because that seems very unreliable. Okay, well, according to Khan Academy, there are only 2 molecules of DNA polymerase 3 present during this entire thing, and one works on the leading strand and one works on the lagging strand. So yes, it's not in a metaphoric sense, it is jumping. So it ends its replication and then it jumps to the next one, ends its replication to the primer, jumps to the next one, ends its replication to the primer, jumps to the next one. There you go. Sorry. Just ignore. You didn't see that. Sorry, that completely... Yeah, just give me a second, guys. I need to be a mod, I guess. Sorry. Okay, anyway. I'm back. Okay. Where is this? What are we saying? When explaining an exam, can we say this? Sorry. One DNA will finish its jump to the next RNA fragment, finishes us there, and then keep going. Yes, you can say that. You can say that. You're fine. To say that, I think. Okay. If you have more questions, feel free to ask. I will stay on this slide for a couple minutes if there's more questions. And until then, I will eat my onion rings in peace. Chili. It's just chili sauce. I can't say sweet or sour. It's Indonesian chili sauce. No, I'm just staying on the slide if there's any questions. I'm just waiting a minute to see if there's any questions. And then we're going to talk about base mismatches. And then we'll take an actual break. I'm just holding in case there's any questions in understanding and stuff like that. Because I know this is very tricky. Yeah, go for it. I can check your understanding if you want to type it out. The reason I said I'm eating is because I'm literally just eating while, people try to make sure that they understand. Very. Very. A lot of it you don't even see because it gets filtered out automatically, but because this is a stage chat, it doesn't get filtered. Yeah, you got bad. Well, at least he put spoilers, I guess. True. Essay incoming. Truly. Actually, we should do that. Okay. What I'm going to have all of you do is... No, actually, I'll make you do that after I talk about base mismatches. Never mind. Ignore me. Foreshadowing. Literally. Sorry for the trauma, guys. Just pretend you never saw it, I guess. Okay, good. If you didn't see it, you didn't see it. Loki, you're better off if you didn't see it. Luckily, when I record when I'm recording sessions, I don't actually... No. Because I don't record this screen. I record my slides. So you can't see it in the recording. Don't all that. It's good that it's not in the recording. Anyone who's watching this anyone who will be watching this recording, hi everyone who's watching this recording, you will not know what happened. It was just a This is gonna be so weird for anyone who's actually watching this recording. It's so funny. Okay, I'm waiting for Gamer75431 to write his response, and then we will move on. Recordings are usually pinned in the chat, so it should be in the N20. Like, any recording that we've ever done is all in the same. Okay, someone else explained. I will read this. Yeah, hi Mama TV. Hi everyone who's watching this recording. Okay, let me read both. This is a question. Each Okazaki fragment is processed by one DNA polymerase 3 followed by one DNA polymerase 1, but across the whole lagging strand, many polymerases 3 and 1 work in sequence or on different fragments in parallel, not just... All you need to know is that it could very well just be one polymerase 3 and one polymerase 1 working on the entire thing. Okay, so the two things unwind, and then helicase is the thing you're doing the unwinding. Yes. Then there is two strands, the leading and the lagging strand. I think that says leading. Leading strand is able to have DNA polymerase add a primer and allows for DNA polymerase 3 to work in the 5' to 3' direction, while on the lagging strand, which goes 3' to 5', yes, we have DNA polymerase 3 add on, but the sense for DNA polymerase have to keep being added, and then polymerase keeps adding, and the RNA is then removed, and polymerase 3 keeps filling in. Okay, so the gaps are filled by polymerase 1, not polymerase 3, but otherwise that is a good enough summary, I think. Oh, and you should talk about ligate. Sorry, no, the gaps are filled. Oh my god, okay, no. The RNA bases are removed by polymerase 1, and the gaps are filled by ligase. Apologies. I realized I said it wrong. Anyway. Okay, so really quickly, we're going to talk about base mismatches. Okay. That says DNA. That's not supposed to do that. It's DNA polymerase 3. Wow. RIP. Anyway. Okay. Fixed. DNA polymerase 3 usually adds nucleotides correctly, according to the rules of complementary base pairing, but occasionally there can sometimes be an error. For example, a free nucleotide with the base G might be paired with T in the template strand, but it doesn't happen very often. It happens every once in a while, just because of normal errors. But then, because our body's really smart, DNA polymerase 3 can recognize a base mismatch between the last nucleotide that it added and the base it added it to on the template strand. What it'll do is it'll excise the incorrect nucleotide, move back along the template strand by one nucleotide, and then reinsert the right nucleotide. This process is known as DNA proofreading, and this is important to know because it reduces the frequency of mutation during replication. So it can be a question: what feature in our bodies, or what enzyme, or not even what enzyme, what feature helps reduce the frequency of mutation? Or just like what reduces the frequency of mutations, and then you have to say DNA proofreading and then talk about how DNA proofreading works. Does everyone understand what DNA proofreading is? Essentially what happens is polymerase 3 will usually be able to identify a base mismatch. It'll remove the wrong nucleotide, move back across the template strand, add the right nucleotide, and keep going as if nothing went wrong. Does that make sense? Yes? Yep. Okay, perfect. So that is the end of DNA replication. But before we mark a full end of DNA replication, what I want everyone to do, I'm gonna move to the next slide just so it's not an actual break, guys. It's not a break yet. Actually, I'm not gonna move to the next slide. I'm just gonna leave it here. We're gonna talk about I want everyone to write a response to the following question, which I'm going to put in chat. And write it as if you were writing it in actually, I'm gonna get the SLs to come back first, and then we're gonna hold on, give me a second. At M225 biology. Okay. Yes. So I'm gonna put a question in chat for both SL and HL. I mean, it's gonna be different questions for SL and H. Oh my god, that's so cute. Wait, I'm literally screenshotting that. That's going on my, like, I don't even know where it's going, but I'm screenshotting that. Okay. Okay, so it's the same question, but it's different marks for SL and HL. The 4 is for SL, the 8 is for HL. It's so cute. I love it. I've screenshotted it. I love that. Thank you. Okay, I've put the question in chat. Everyone has to answer it. Let me stop screen sharing. Everyone has to answer that question. Yes, you have. I will give you guys. What's a realistic amount of time to give you for an 8? Okay, the SLs should be able to do it in 6 minutes, and the HLs should be able to do it in 10 minutes. No, I'm gonna do 5 minutes and 10 minutes. There you go. So you have 5 minutes / 10 minutes. If you take longer than that, I'm not gonna punish you, but I'm just like, yeah, realistically. Let's just, yeah, okay.